Consider supporting Archaeo-Soup on Patreon for as little as $1 per month. Link available in the video description. Thank you.
Hello, Ali Poulter.
Hi, Mark. Nice to meet you.
You too, you too. Now, I have reached out to you. Well, actually, indirectly, I suppose I reached out to your team, your publishing team, having come across a very interesting article on sciencealert.com. And this particular thing is very close to my heart, I suppose very close to my head, in some ways. And before we get into it, I'd just like to possibly ask you to introduce yourself, obviously who you are, where you are, and what you do.
Yeah, sure. My name is Ali, or Alexandra Poulter. I'm a master's student at UCL in the Institute of Archaeology. I'm studying bioarchaeology, but I also work as an undertaker and embalmer at night, and as a conservator at Bart's Pathology Museum during the day. And I'm currently researching the preservation of brain tissue in archaeological contexts.
Well, goodness gracious me, you just went like through the roof in terms of coolness, didn't you? Sherlock Holmes is alma mater, and you have a nighttime job as well. It's very impressive. It's like a superhero of the grim. Yeah, it's cool. So we're talking, or rather, I've invited you today to talk about the Heslington Brain.
Yeah.
And this is something that I really should have spotted earlier, in so much as I was actually at the York Archaeological Trust when they found this thing, and I saw it in the lab, and essentially it's a 2,600-year-old chunk of brain that's now 20% of its original mass, because it sort of lost moisture, I think. And I suppose the question is, and the question in this article, and the question I'd like to talk about today is, well, I suppose first of all, how on earth did this brain survive so long? There are other questions, but the first one is how? Because normally it's stone, bone, and pottery, isn't it, that archaeologists are dealing with?
That's it exactly, and you've hit the nail on the head there, Mark. There's an unfortunate tendency in archaeology to dismiss anything that isn't bone or stone as somehow outside of our purview. So this paper that you mentioned, led by Dr. Axel Petzold, a colleague at UCL in the Institute of Neurology, is just so exciting because soft tissue preservation, I mean, we don't expect to find a brain that's 2,600 years old preserved.
No.
So what my colleagues have theorized is that the brain is an exceptionally unusual organ. It's mostly water, about 80%. And you mentioned that when we find these brains, they're shrunken to about a fifth of their volume. Well, the remaining 20% of brain is roughly 50 fats and proteins. And again, that's very unusual. Most organs aren't 50-50 fats and proteins.
No.
But to maintain its hundreds of trillions of synaptic connections, the brain is reliant upon the structural stability conferred by a matrix of proteins called intermediate filaments. And what Axel and his team have theorized is that these filaments in themselves are also unusual. They are inherently unstructured. They have large polyanion tails, multiple phosphorylation sites, and they can self-assemble into polymers, and they form both intra and extracellular aggregates in pathological conditions. And Axel and his team have theorized that it's this protein aggregate formation that might be permitting the Heslington brain and perhaps others to persist through time.
Okay. And so would it be an appropriate analogy to look at, say, wood in the same way that the collagen material in wood, often often the living stuff in wood goes away, but the structure is preserved. Often we find it in like a wet setting, so the water has filled up those structures, and we have to replace that water with a form of plastic to keep its structure. Is it sort of almost like the skeleton of the thing, of the brain, is what's sort of what we're seeing, and the certain chemical processes allow that skeleton to form into slightly different, almost well, for different structures, I suppose. I'm trying to find a way of describing this that isn't too jargony, if that makes sense.
No, I
Hmm.
appreciate that. It's we are talking about the chemistry, and we
Yeah.
have to respect the chemistry. But I appreciate where you're coming from. That
Hmm.
it can get a little jargony.
No, it's fine, it's fine. I just thought I'd just attempt to to break it down a little bit.
Well, I'll do my best to
Yeah,
break it
yeah.
down as well.
Please.
The truth is, we don't know. We don't know why these brains are preserving.
Okay.
What I would say is a recent paper was published by Petrone and colleagues, and they helpfully introduced the concept of vitrification of ancient biomolecules. Vitrification is underpinned by studies of anhydrobiosis,
Okay.
which is a very low energy metabolic state that enables organisms to persist in a kind of condition of suspended animation.
Hmm.
And what that means is that as the proteins are dehydrating, sugars typically replace the water. And we see this in wood, for example. This science has also been used to develop products that form glass in the presence of macromolecules. So while I think that vitrification could be a really interesting take, and it might be explaining the preservation of some ancient biomolecules, whether or not that's happening in the brain, we just don't know.
Interesting. Okay. Well, actually, that is interesting in so much as so. Yeah. So as you say, it's a possible analogy, but also it doesn't actually describe or explain what we're seeing. That okay. Cool. I like that. So. I suppose, how often do you think this might happen? Because I remember the reason why I asked that is that I remember someone at the time saying this was early days, obviously, when this had been identified, suggesting that on many excavation sites you'll have, in particular, students or maybe volunteer diggers who are clearing out what they recognise as a bone, a very geometrically sound and recognisable form, and in there may well just look like material, worm
Yeah.
feces and soil, and sort of you might get that slightly bubbly material that comes out of living soil, but you're not
Yeah.
that could look a lot like brains. So people might have been clearing out brains for decades and not knowing
I,
it.
I'm afraid I think that's probably the case.
Right.
As you mentioned, there's very little awareness amongst an enormous proportion of our colleagues for the potential for brain tissue to preserve in the
Hmm.
first place, let alone the vast numbers at which it patently does so.
Right.
My colleague Sonia O'Connor, who's at the YAT, and who wrote the 2011, the original report on the Heslington Brain with a vast number of colleagues and collaborators, initially found 300 reports of brain preserved in dating back to 1960.
Okay.
I've critically reviewed all of those papers, and I've subsequently found more than 1,000 further brains in reports dating back to 1651.
Wow.
So it has been
Yeah.
known since the 17th century that brain can preserve, but I'm afraid that it's still when brain tissue is found, there's this unfortunate tendency to kind of treat it with a reverence.
Yeah.
That these samples are touted as unique and
Yeah.
one of a kind, and perversely that often means they're minimally investigated.
Okay.
As you mentioned, I'm pretty sure that a lot are getting washed out as field archaeologists are washing the skeletons.
Hmm.
And I would just love to put a shout out there. Please be aware of the potential for brain to preserve, and think very carefully about what you might be cleaning in terms of intracranial material.
Yes, absolutely. That should be on a T-shirt.
Exactly, yeah.
And so, I suppose the reverence with which these brains are held, that's going to be a byproduct of sort of cultural connotations with the importance of the brain for self and thinking and this kind
Yeah.
of thing. But if then there are records going back to the 17th century, you said 1600s, why aren't they more commonly recognised in basic excavation teaching? For example, I've never once prior to then had never once been asked or told about this possibility, and even now I very rarely hear people As you say, keeping it in mind, pun
Yeah.
intended, I suppose.
No.
So,
Yeah.
so
Oh
what?
my goodness, the world is fun when you get into brain
I
preservation.
can imagine. So, but why do you think then it isn't more well known about? I suppose, and to yeah, put it simply.
Do you know, I'm not sure I have a good answer for that,
Hmm.
Mark. Again, I just want to mention my colleague Sonia O'Connor. She is instrumental in this entire field. If it weren't for Sonia and her work with the Heslington, and actually it was because Sonia had told her students, be aware that brain can preserve, that the Heslington brain was found.
Right.
And the Heslington is an exceptionally well-preserved example.
Hmm.
But there are others. And while brain tissue, I wouldn't call it common, it's certainly ubiquitous. It's found all over the world in wet and dry climates, in hot and cold climates, in acidic and alkaline soils, aerobic and anaerobic conditions. So why it's not more commonly recognized, I'm not sure I have a good answer. My only feeling is that, again, I mentioned that it's often minimally investigated, and I feel that might be partly a function of the seemingly eccentric nature of the material,
Okay.
and this kind of tendency to for a quick eye-catching paper. And what I and my colleagues are calling for is a radical and conscious rethink in that respect. Let's stop treating this material as exceptional and unique and one of a kind, and instead really focus on in-depth, comprehensive, systematic investigation. And hopefully we can make this more widely known about.
Okay.
And obviously the more we talk about it, the more we publish, the more research we do, the more brains will be found.
Yeah. So, so in that case, then, if the brain one day was seen, or in the very near future is seen as likely to be found in a grave as a skeleton, what comes out of that in terms of investigations of individual bodies, and for non-metric variations and all this kind of thing? And I suppose I'm asking two questions, and so much as, first of all, what can we tell about a brain? Why should we care, or why should anyone at home, as it were, care about finding brains, as opposed to, say, a skeleton can tell you about height, or can tell you about some diseases, so on and so forth. And then so that question, and then I suppose attached to that is, what is the potential once we care about it? What is the potential for this sort of comparative brain study? Is there likely to be the sort of population analyses that we can do with skeletons and between populations and so on and so forth? What do you think?
I'll answer the second question first, if I may,
Yeah.
Mark. I'm not sure that we'll ever be able to tell anything on a population scale from preserved brains, for one important reason, which is, say you have a cemetery of 100 individuals.
Yeah.
Maybe, just as a ballpark figure, say 10 of those brains preserve. They're not all going to be in the burials that are next to each other. They might be dotted all over that cemetery, and we don't know why one person's brain preserves and why another person's doesn't, which I think is incredibly a fascinating question that I can't wait to explore further and answer. So I'm not sure we'll ever be able to say anything on a really population level, but individually, oh my goodness, I think that this material could be incredibly rich in terms of its interpretive bioarchaeological value.
Okay.
For two reasons. One, the biomolecular data that we may recover, and also the architectural data. In terms of the biomolecular data, I'm thinking especially of proteins. At the moment, we have very limited objective methods for studying things like brain aging, brain development, neurodevelopmental abnormalities, neurodegenerative disease, or mental health in the past. Clinical neuroscience relies on empirical quantitation of biomarkers, protein biomarkers, in the case of neurodegenerative diseases like Alzheimer's, Parkinson's, Huntington's, or other dementias. I'm thinking of alpha-synuclein, amyloid beta, phosphorylated tau, these sorts of things. And the paper that we spoke about initially at the start of this conversation, by Dr. Axel Petzold and colleagues, have recently recovered more than 800 proteins from the Heslington Brain. That is by far the largest ancient proteome recovered from any archaeological material ever investigated to date.
Right.
So this would be an incredibly rich resource.
And just have interest in it. Does that include comparisons with, for example, you hear about things like baby mammoth recovered from permafrost? Does that include that in terms of protein recovery? Or
I'm not aware of a proteomic investigation
okay.
of mammoth brains. Actually, having said that, I think there might be one by possibly Krebs and colleagues in 2016. But again, that's several years ago, and the
Yes.
technology is moving on so quickly.
Mm-hmm.
I think what's especially innovative about Axel's approach is that he's used a gel-based extraction, which seems to have been especially effective. And I'm really looking forward to interrogating his data in more detail. But I think that what that will enable us to do is both compare the proteomes from different individuals within a single assemblage and also across archaeological sites. A paper by Maixner and colleagues in 2013 was, I think, the first to reveal protein biomarkers from an ancient brain. This is Otzi the Iceman, that are specifically associated with certain pathogenic and metabolic pathways of neurodegenerative diseases. I just think that's incredibly valuable in order for it to have an objective exploration of these things in the past. If we can quantify, oh my goodness, the possibilities are endless.
So just for clarity, the signal went a bit dodgy there. That was Otzi the Iceman you were mentioning there. Yeah. And so I suppose would this lead to modes of understanding that lead to forms of treatment, or is it a matter of understanding the history of different types of brains and different types of brain degeneration as well, can lead to in a more roundabout way, can give a context for medical understanding? I suppose, to put it another way, is it the hunt for the cure for Alzheimer's, or is it more actually about, and I can sort of tell from the is it more actually about the context of these things in terms of the human condition? Because for example, I have a friend and colleague who very much likes her leprosy, as it were. Yeah, don't we all? But what's interesting is that her study of leprosy has very direct connotations for origins of particular microbes that cause these sorts of problems. So is that what you're after, or is it more about, as I say, the general sort of is it big data basically that you're after in essence?
I think that's fascinating. I'd love to hear more about your colleagues' work in leprosy. What I think that there are a number of different fields that the study of ancient brain could both be informed by and also impact. I'm thinking of things like clinical biomarker research, but also paleoneuropathology, evolutionary anthropology, structural and functional proteomics. I mean, it would be fantastic if there were implications for modern treatments, but for me, at least, the idea of being able to explore not just neurodegeneration or neurodevelopmental abnormalities, for example, through time, but the broader experiences of learning and brain aging, for example. I mentioned that I think that there are both implications for the biomolecular richness as well as an architectural richness, if you like. If brains are preserving in the way that Axel and his colleagues and others have theorized, as you mentioned, they're shrinking and they're getting smaller and smaller over time, but it's feasible that that shrinkage is isotropic, in which case maybe the neural network is being preserved, in which case you can ask questions that we can address through neuroimaging, like, could this person long dead read and write? Imagine if we could study things like literacy and numeracy in the past.
Mm-hmm.
Relatedly, the Lundbeck Foundation Geogenetic Center in Copenhagen are sequencing 5,000 ancient human genomes to explore the origins of our mental health through time.
Right.
So people are already working on these things. And if we can put proteomic, genomic, and architectural investigations together and come up with a kind of holistic characterization, I think the possibilities are incredible.
Yeah, I can imagine. Well, indeed, I can imagine, arguably because of these structures, precisely. So, so just to be clear, then, when you talk about the possibilities of understanding whether someone the extent of their literacy, is this because of formations in the brain linked with learning about reading, writing, and comprehension, so on and so forth?
That's it, exactly. So my undergraduate degree and the reason I came into brain research is it's in neurolinguistics. I studied Arabic, Latin, Old English, and I became interested in the neuroanatomical basis for language acquisition. And whereas we have specific brain regions that have evolved over time to accommodate spoken language, reading and writing is not a natural phenomenon in the sense that we haven't
It's very
evolved
abstract,
to
isn't it?
do that. Yeah. So what happens when we learn to read and write is there are certain neuroanatomical changes that take place in the brain. And they are very particular. They occur at the same brain coordinates in all individuals within a few millimeters of each other.
Right.
So that if you use, for example, diffusion-weighted MRI, which is a technique to map the white matter pathways in the brain, you can tell based on the course that those white matter pathways take whether this person can read and write and whether this person can't, for example. The brains look very different. And if the neural architecture is being preserved in these ancient brains, we might be able to tell the same things.
Okay, that's a level of specificity, which is as it were, an order of specificity that goes that has far more practical applications than, for example, in the past I've heard people talk about how humans have the FOXP2 gene, this kind of thing, sort of post Homo erectus or something, as a potential for spoken word. But that doesn't tell you much about, because then the problem is once you start to try and recreate formations and talk about palate use and tongues and throats, it's very difficult. Whereas actually, the ability to actually have the soft thing not only there to observe but also tell you something so specific is very incredible. And also, the implications are immense in terms of, because it may well be that there are. You could imply forms of writing and language that go back further than obviously written word survives. That's very exciting. Yes,
And the more we talk about it, the better. The more people are aware of the implications, the better. So.
Yeah, absolutely. Okay. Well, it's interesting because I suppose one of the follow-on questions I thought I would be asking you was going to be along the lines of whether essentially a more literal form of the way in which the constructs of the brain form over time. I'm thinking about, for example, this idea of the cathedral mind. This idea that you can see how, also theoretically, obviously it took a long time for neuroscientists and archaeologists to start talking to each other. But the idea that that portions of the brain almost as structures add to the capabilities of the creature. Leads one to immediately think and ask whether or not that you could make a comparison between, say, a 500-year-old brain and a modern brain, and come up with any worthwhile questions or comparisons or contrasts. Is that likely, or are these structures formed over much greater distances of time? Because and I know I'm so rambling here, but I should explain the reason why I'm asking that very specifically is because we can infer an awful lot from environmental changes. For example, on the one hand, say a Tudor person living in London might have had, shocking state of health in some ways compared to a modern person. But in other ways, a modern person is much more, especially in the Western world, exposed to much more insidious and low-level toxins and things like that than a Tudor person would be. Is there anything like that that might come out of this, or as I say, is it much more of like a 10,000, 100,000 year development that you're looking at?
Wow. Well, I mean, I would love to think that that is possible, and I certainly am. I'm going to keep an open mind, and until we work out how exactly these brains are preserving, it's a difficult question to answer.
Right.
The oldest preserved human brain tissue that I've come across is 8,000 years old.
Okay.
So evolutionarily speaking, that's not a very long time scale. But on the other hand, the brain is exceptionally plastic. It changes in all of us through time. It changes constantly as we learn new skills and acquire new experiences in our lives. So whether or not we can infer anything on an evolutionary scale, I'm not. My instinct is no, but let's keep our fingers crossed and see what we can come up with. But certainly, again, on an individual basis, I think if brains are preserving in this way and the neural architecture is being preserved, clinical neuroscience is constantly evolving, and the amount of information that we can glean from the diversity of, for example, white matter pathways is just extraordinary. For example, we can tell clinically whether someone has had a deprived childhood. Imagine if we could do that in the past. That would be extraordinary.
Yeah, absolutely. Okay.
Actually, something that does come to mind, I'm sorry to interrupt you there, was I am aware of one previous investigation that has looked at imaging the neural network of preserved brains. That was a study by Burns and Ashwell in 2017, and what they did was image an extinct Tasmanian tiger alongside a modern Tasmanian devil to look at the differences between how the brains may have developed over time. So it's possible.
Okay. Okay. Interesting. And I suppose just one more related question to that then would be, would there be any scope for relating these sorts of observations about brains that are within that window of preservation? Let's say 8,000 years is the top of that. The sort of the soft structures, and comparing that to how they relate to the hard structures. For example, you sometimes see obviously swirls and whirls on the inside of skulls that relate to brain formation. Is there any scope there to sort of say compare, I don't know, a human brain that's as old as usefully observable to, say, a Neanderthal skull, and come up with meaningful comparisons in terms of formations of particular structures within the brain? Or again, is there just too much gap between the exterior of the, say, Neanderthal, the interior of the skull and the interior of the brain to actually come up with a meaningful comparison? I know I'm probably asking you very hypothetical questions here, but I'm just intrigued in terms of the possibilities, if that's what I mean.
Yeah. Well, you're right. It is hypothetical at present until we get some more data and do some more research. But it's an interesting question. It's something I've wondered for a long time. I mentioned that my background is in neurolinguistics, and when we're looking at the evolution of the human brain, often what we're reliant upon are fossil endocasts. And I've often wondered about, I mean, there has been work, obviously, in whether we can infer much from the interior of the cranium, but whether we'd be able to correlate the two, of, say, a preserved soft brain with the cranium that it's associated with, I'm not sure. It'd be interesting to find out, and it would certainly have implications for all the previous studies that have come before us in paleoneurology that are reliant on inferences based on those endocasts. It would certainly be interesting. I'd love to know if there are certain differences in brain structures, like Broca's and Wernicke's areas, for example, which are associated with speech production and comprehension. There have been a number of different investigations to see how they might have changed over time as we in theory become more fluent and well spoken as human beings, but we'll see.
Yeah, okay. And I suppose more or less finally, I've been trying to find something now for years, and you may know what this is.
Oh,
I
okay.
remember when I was first studying human evolution, someone suggested to me that there's a beach somewhere where there are brain stones, because the beach once had a whole host of monkeys die there, like millions of years ago, and their skulls filled up with silt, the bone eroded, and the brain was essentially cast inside the skulls. Do you know where that is? Like, I'm trying to remember where it is, and I can't.
I've not heard of that. So now the
Okay.
pair of us are going to spend the rest of our lives trying to find out where
Yeah.
this is. I've not heard of that, Mark.
Okay, it's
If
driving
you
me
find
mad.
out, let me know.
Yes, if anyone at home knows, please comment below. It's literally
Yes.
driving me potty.
Leave a comment.
Yeah. Well, thank you very much. Thank you. It's been a real pleasure. And actually, as ever with these conversations, they often go off in directions that I don't entirely predict, based upon good answers. So thank you. And I suppose finally, as I'm almost always suggest in these conversations, is there anything that you would like to put out there in terms of something that you want more people to know about this particular field of study? I mean, obviously we've touched on the potential for finding brains, but is there something that people always say, or is there some sort of cliche, maybe even that I've come up with during our interview, that you'd like to dispel or challenge, or just an interesting fact?
I would absolutely. The key point I really wanted to make, I made earlier about, if you find any brain tissue, if you're even 5% certain that you've got brain, just be careful with it. Let me know. Leave a comment in this video. Please tell someone, because we need to be finding more of this tissue. It is ubiquitous across the world and across vast swathes of time. One bugbear I have, you mentioned if there are any kind of myths, is that often this material is investigated in such a cursory way, and there is an unfortunate tendency to attribute its preservation to adipocere formation.
Okay.
Adipocere is a word that's bandied about in bioarchaeology and forensic anthropology, and there are various number of reasons why it's highly unlikely that brains preserve through adipocere formation or saponification. So that is something I want to correct, and I hope my research does that in the next few years.
Okay, cool. And just in case people don't know what those words mean, myself included, what are those forms of preservation? And I suppose, why do people think that they're so common?
Adipocere formation is also called grave wax. So it's
Okay,
waxy, the texture varies actually depending on the ionic involvement, but it tends to be crumbly, waxy, obviously, and it forms readily from lipids, but specifically trace or glycerols. And the brain is only a very few percent trace or glycerols. It also increases the volume of affected soft tissue, and as we've discussed, brain tissue is invariably shrunken when we find it. But adipocere formation is quite common. It's very bizarre when you see it on a corpse. But yeah.
Okay, thank you very much. Thank you. I won't Google it.
I almost added don't Google image facts. It's not very nice.
Thank you very much.
Thank you, Mark.
Well, if it's okay, if anything more solid, as it were, comes out of this in terms of interesting ideas, that as opposed to me just hurling hypotheticals at you, I would love to revisit this in the future.
Absolutely, so would I.
Grand,
yeah.
Wonderful.
That would be great.
Thank you very much for your time.
Thank you. Mark.
Okay.
Take care.
Bye
Bye.